Hello, good evening. I'm audible. Hello, everyone. Good evening. Am I audible? My voice is clear. Good evening, Vijay. Good evening. I hope I'm audible clearly to all of you. So, today we will start discussing about the quick revisions in this marathon series. Today we are the last part of this marathon series that is comes under your other subject. Okay. So in this video, we are going to discuss about 100 important MCQs that is related to your other subjects. Okay. So before starting this session, so let me tell you something. So as we are in the last part of our preparation, so I want to tell a simple line and I will start my class. So there is a beautiful saying by our one of the renowned and late, we can say our a person of our India, Sir Ratan Tata. So he told that all of us does not have the same talent, yet all of us should have a equal opportunity to increase our talent. So what does it means? So when we are born, we don't have the equal talent, but when with the time, with the when we get the more opportunity, we get the increase our talent. So today in this platform, GDC has given you a beautiful opportunity to make your preparation more clear, more better, and you prepare all of you to a great level. So that's all for my saying. I hope this is audible to all of you. Let's start. So in this scenario, in this class, we are going to discuss about your basically biochemistry, biotechnology, microbiology, MCQs. Let's see the first question. So I will show the question. So you have to give the answer. Later, I will explain what is the explanation behind that particular answer. So first question. Which of the following is a sugar alcohol? Your option is glucose, stearic acid, xylitol, yeah, sucrose. So first thing is what is sugar alcohol? So when we talk about sugar or carbohydrates, so these are basically consisting of polyhydroxyaldehyde or ketone. So they are basically consisting of polyhydroxyaldehyde or ketones. But in the sugar alcohol, instead of multiple aldehyde ketone group, you will find this hydroxyl group. Okay. So which of the following is a sugar alcohol? Your option is glucose, stearic acid, xylitol, and sucrose. Okay. All of you try to answer the question. So the correct answer here is xylitol. Why xylitol? Let me explain it. Xylitol, sugar alcohol, these are the type of carbohydrate that is derived from the sugar and have a similar chemical structure, but they are not fully absorbed by the body. So they are used as a sugar substituent. They are called as sugar alcohol, naturally found in the fruits and vegetables. It is known for its sweetness and uses sugar-free product. So most important thing about sugar-free alcohol is they are basically low calorie. So when you eat the sugar alcohol, you will get a less calorie as comparison to the other sugar. Okay, very good. Uh, Rajesh Patil have given it has OL at the end. Very good. Nice trick to remember. Next question. DNA amplification by polymerase chain reaction uses what? So this is basically we are talking here about the question related to PCR. So when we talk about PCR, so PCR is basically nothing but this is a cell free amplification technique. That means without involving a cell, it can amplify in your system. So in this step, so DNA amplification by polymerase chain reaction uses which enzyme? Thermus aquaticus DNA polymerase, DNA topoisomerase, RNA polymerase, या RNA helicase? Yes, all of you, please give the answer. Very good, very good. So here the correct answer is DNA polymerase, that is Thermus aquaticus, or also called as Taq polymerase. It is also called as Taq polymerase. Okay, it is called as Taq polymerase. So let's see what are the other Taq polymerase are used in this technique. So this PCR technique is developed by a scientist named Gary Mullis. So PCR basically this is a thermocyclic technique for the generating large quantity of specified DNA. Okay, source of DNA polymerase includes, so basically it's the source is Thermus aquaticus. Many times this question have asked Thermus aquaticus. Okay, so what are the other uh, DNA polymerase that are used? The list I have given here. PFU polymerase. This is for use for the cloning, mutagenesis, sequencing application. Wnt polymerase for high field PCR. Fusin polymerase used for accurate amplification of long and high GC content DNA. Q5 polymerase for cloning, sequencing, site-directed mutagenesis. Hot start polymerase used for multiplex PCR and diagnostic PCR. Long range PCR polymerase used for the whole gene or genomic region application. Okay. So moving to the next question. Deficiency of magnesium usually leads to growth retardation, mental disease, Parkinson's disease, yeah, neuromuscular. Yes, answer the question. Deficiency of magnesium usually leads to which of the following causes? Growth retardation, mental disease, Parkinson, and neuromuscular. So when I was talking about this nutrients, so I told you nutrients are basically two type, either macronutrients or micronutrients. So macronutrients are those which required in higher amount as comparison to micronutrients. So here the magnesium. So magnesium basically comes under your macronutrients. Okay, magnesium is comes under the macronutrient. And this magnesium is basically deficiency leads to neuromuscular weakness. Okay, so the correct answer, yes. Correct answer is option number D, neuromuscular. Please read the question very carefully. Sana, very good. Correct answer. Those who have given the option number D, very good. The correct answer is D. So let's see. So this magnesium, so major constituent of magnesium is basically present in your bone, teeth, cofactor for enzymes such as your kinase. So its deficiency leads to neuromuscular weakness, irritation, and sometimes it leads to convulsion. Even it can lead to epilepsy-like conditions. Okay. So it is daily recommended dose is 300 to 350 mg per deciliter. And the major sources includes your cereals, vegetables, fruits, and milk. So if we talk about the classification of minerals, so minerals are two type, either they are macronutrients or they are micronutrients. So macronutrients are basically your calcium, phosphorus, magnesium, sodium, potassium, chloride, and sulfur. In the question is about magnesium. And its magnesium deficiency is basically associated with which disease? It is basically associated with your neuromuscular, neuromuscular weakness. Okay, very good. So moving to the next question. vaccine provide which type of immunity? Okay, so vaccine basically provide which type of immunity? When we will talk about the immunity, so immunity are basically, or immunology or immunity are basically of two type. One is your acquired, okay, another one is your innate. So innate means this is a immunity which is present since our birth, just like your IgM. If we'll give an example, IgM immunoglobulin. IgM immunoglobulin basically transferred from the mother to the fetus through the placenta. Okay, that is basically provides a immunity to the offspring. When we talk about acquired immunity, so acquired immunity is basically developed over a period of time. And vaccine is basically given for a particular disease. So suppose a person is having tuberculosis, or for the tuberculosis BCG vaccine is given. So that BCG vaccine is basically going to provide immunity for the tuberculosis. That immunity is not present since the birth. It is basically developed throughout the time. So vaccine is comes under your acquired immunity. Option A is the correct answer. Very good, Abhishek. Very good, Darshan. Very good. Not both A and B. No, both A and B. No. Innate immunity is basically. Please listen my words carefully. Innate immunity is basically there since our birth, and this is due to the mother's IgM immunoglobulin that is transport from the mother to fetus to the placenta. Okay, so you can see vaccine is biological preparation. This is provides the acquired immunity to a particular disease. It contains agent resembles the disease causing microorganism. They the substance basically used to stimulate or basically production of antibodies and provides a immunity against several disease conditions. Okay, so moving to the next question. In which method an order of a fixed number of item is placed every time and inventory levels are to a predetermined point. So question is, in which method we basically fixed a number of item is placed every time and inventory level falls to a predetermined point. So what is basically called? ABC method, maximum and minimum method, open to buy method, yeah, economic order quantity. So this is basically comes under a economic order quantity. So what is basically a economic order quantity? Like option D is the correct answer. Let me explain you why it is economic order quantity. So economic order quantity, it is basically a mathematical tool. We basically use a mathematics to determine the order quantity that minimize the cost of ordering and holding the inventory. Okay, so economic order quantity, this is basically a mathematical calculation which basically determine the order quantity which will minimize the cost of ordering and holding the inventory. For suppose I have 5 packet of particular drugs. So I will calculate that yes, this 5 packet or 5 strip of the drug, how much time it will run. So on the basis of my mathematical calculation, we will give the further order. So that is basically called as economic order quantity. Okay, very good. Economic order quantity, very good. Option D is very good, correct. Yes. Now, moving to the next question. Number of mole of ATP produced when one molecule of glucose is completely oxidized. Okay. Okay. So when one molecule of a glucose which completely oxidized, how much ATP will be produced? So it is talking about oxidized means presence of oxygen. So we have two scenario. One is your aerobic oxidation, aerobic condition. Another one is your anaerobic condition. In aerobic condition, the total ATP that is produced is 38. Okay. In case of anaerobic condition, the total ATP produced is, yes, yes, answer me this question. In anaerobic condition, how much ATP will be produced? In absence of oxygen, how many ATP will be produced? Yes, answer this question. Abhishek, zero. No, not zero. How much ATP will be produced during anaerobic condition? 2 ATP. Very good. 2 ATP. So 2 ATP will be produced in anaerobic condition. Many times this question I've asked. You have to remember in aerobic condition, 38 ATP will be produced. In anaerobic condition, 2 ATP will be produced. So from glycolysis, total ATP is produced 8. Okay. From TCA cycle, okay. So by combining your glycolysis and your TCA cycle, total 38 ATP will be produced. Okay, in anaerobic condition, total 2 ATP will be produced. Okay, please remember this question. Next question. During the denaturation of cycle of PCR, what is the temperature? So if we'll talk about the PCR, so PCR basically done in 3 steps. One is your denaturation, annealing, and third one is, so denaturation step is carried out at 90 to 98 degrees centigrade. Annealing is carried out 50 to 55 degrees centigrade. And synthesis is carried out 72 to 75 degree centigrade. Please remember this temperature very carefully. Okay. 90 to 98 degrees centigrade. Option A is the option A is the correct answer. Okay. So in denaturation, the temperature is around 95 degree. In annealing, the temperature is 55 degree. In extension, temperature is 75 degree. Denaturation is heating step, annealing is cooling step, and renaturation or extension, or this is also called as synthesis. This is called as heating phase. Okay. So moving to the next question. Positive Benedict test is not given by. Positive Benedict test is not given by. Who can answer this question? So if you remember, this positive genetic test is not given by. So positive genetic test basically given by, genetic test is given by for the non-reducing sugar. Okay, this is for the non-reducing sugar. So here Which of the following is a non-reducing sugar? Sucrose. Okay, option A is the correct answer. Very good. All of you have given the correct answer. Please share this class link to your friends so that all of your friends can be benefited from this discussion. So sucrose is basically non-reducing sugar, so that's why it will not give Benedict test as well as Barcode test. Okay, so this is the list I have kept here. So which are the test, which are the molecule which will give the Benedict test? The monosaccharide that will give positive for Benedict test includes glucose, fructose, galactose, ribose, and xylose. And the polysaccharide which will give a Benedict test is your lactose and maltose. Next question. COMS test is used for the detection of. So COMS test is used for the detection of atypical antibodies, diphtheria antigen, anti-RH antibody, and atypical antigen. This COMS test is basically used. Yes, answer this question. So the COM test is basically used for the detection of anti-RH antibody. It is used for the detection of anti-RH antibody. Okay. So if you see the COM test is basically used to detect against surface of a RBC, because you know on the surface of our RBC, on the surface of our RBC, these RH antibodies are there. RH, so RH factor is there. So that will be detected by this COMS test. Okay, that will be detected by COMS test. Moving to the next question. Given each and followings are the source of primary data except which of the following is a source of primary data except? Your options are trade association, salesman, consumers, and dealer. So please understand the question. The question is about which of the following will give the project which of the following will give a data about uh, your primary data. So the correct answer is salesman. Yes, salesman will give the description about primary data. Consumers will be give a description about the primary data. Dealer will be give a description about primary data. But this trade association will not give the prime data about primary source. Okay. So please remember the trade association will not give data about primary. So trade association, this is not typically considered source for the primary data. What is basically primary data means? Primary data refers to the original data collected directly from the individuals or source of first hand of a specific research purpose. So what is telling that this is the data that is originated from the directly from the individuals or sources for specific research purposes. Salesman will provide insight into customer preference. Consumers gathering information directly from the consumer through the survey, interviews, or observations. Dealers information from the dealer or distributor or retailer can be valuable for the understanding market dynamic, demand pattern, as well as product performance. Okay. Moving to the next question. Next is in the citric acid cycle, the conversion of oxalosuccinate to alpha-ketoglutarate is catalyzed by which enzyme? The isocitrate dehydrogenase, succinate dehydrogenase, aconitase, yeah, none of these. So, in this citric acid cycle, the conversion of oxalosuccinate to alpha-ketoglutarate is catalyzed by is catabolized by the enzyme isocitrate dehydrogenase, succinate dehydrogenase, aconitase, and none of these. The correct answer is yes. Answer the correct answer. Okay. Next. So the correct answer is isocitrate dehydrogenase. So there is a simple mnemonic which you can remember. Okay. The mnemonic is Charlie and Isabella always sing song for me. Charlie stands for citrate synthetase and stands for aconitase. Isabella stands for isocitrate dehydrogenase. Always stands for alpha ketoglutarate dehydrogenase, succinate thiokinase, succinate dehydrogenase, fumarate, and malate dehydrogenase. These all are basically the simple mnemonic that is Charlie and Isabella always sing song for me. This is a simple mnemonic which will help to remember the uh, which will help to remember the detailed enzymes. So moving to the next question. Hybridoma technology is basically widely used for producing callus culture, organ culture, monoclonal antibody, attenuated microorganism. You will see what is the use of hybridoma technology. Hybridoma technology is basically used for the production of monoclonal antibody. Please remember monoclonal antibody. Okay, it is used for the production of monoclonal antibody. Hybridoma technology, and this is used basically tissue culture like a callus culture. So basically, this is a fusion technique. So here we use B cells, that is B cells, which is responsible for the production of antibody with a cancerous cells to fuse this both B cells and cancer cells to form a hybrid cell, that is myeloma cell, which will use for the production of monoclonal antibody. Okay. So this is a produce one type of immune cells and clones to a single parent cell. This monoclonal antibody can be produced by specialized cell through the techniques known as hybridoma technology. Okay. So moving to the next question, which of the following is a example of product that developed through medical biotechnology? Okay, which of the following will develop through medical biotechnology? Your options are antibiotic, biofuels, genetically modified crops, industrial engine. So these biotechnology derived products are basically derived from the example is your antibiotics. Okay, antibiotics are basically developed under this medical biotechnology technique. So if we'll see, so this is medical biotechnology techniques are basically used for the development of various pharmaceutical products, including your antibiotics, vaccines, and therapeutic proteins. Okay. So moving to the next question, which of the following immunity is also called as antibody mediated immunity? Okay. So your options are acquired immunity, cell mediated immunity, humoral immunity, innate immunity. So first thing is you remember which immune cells is responsible for the antibody production. If you remember our B lymphocytes, B lymphocytes are or B cells are responsible for the antibody production. For the antibody production. And these B cells are basically forms under which type of immunity? It is forms under humoral It is comes under the humoral immunity. Okay. Humoral immunity is also called as antibody mediated immunity. So this consisting of antibody present in the body like humor, blood, and lymph. Antibody protects our body from the pathogen that enters to the blood or lymph. Okay. So moving to the next question, the key component of total quality management. So what is the key component of TQM? Your options are total control of all quality related activities, commitment for all employees to quality improvement and having team meeting, top management's direct involvement, and the introduction of ISO 9000 series. So, what is the key concept? So, the key concept behind total quality management is your top management's direct involvement. So, how the top managements are involved? Okay, option C is the correct. I hope. So the key concept includes top management leadership, transformation of organizing culture, process focus, employee education training, quality measurement and statistical activity, recognition and reward, creating framework for quality, customer focus, collaborative approaches, benchmarking, employing empowerment and management integration. So moving to the next question, following amino acid does not exhibit optical isomerism. For the optical isomerism, what is important? That is your chiral center. Amino acid should have a chiral center to call it as a optical active. So which amino acid does not have chiral center? Yes, the option is C, that is glycine. Glycine is optically inactive. Okay, its structure is like this. This is the structure of glycine. So it is considered as a optically inactive. Okay. So this question was asked also in GPAT 2020. Moving to the next question, agent that can cut the DNA at a specific sequence in a recombinant DNA technology are called as exonuclease, phages, plasmids, a restriction enzyme. So which agent that cut the DNA at a specific site? Option is exonuclease, phages, plasmid, and restriction enzyme. So the cutting of DNA at a specific site is basically done by restriction endonuclease. The enzyme they called as restriction endonuclease. The technology that is made possible by two type of enzyme, that is restriction endonuclease and ligase. Restriction endonuclease recognize the specific sequence of DNA and cuts within or close that sequence. Cutting and joining together the vector of DNA fragment from different origin result in recombinant DNA. Okay, did recall the restriction endonuclease are used to cut the DNA. And please remember, ligase, one of the ligase is responsible for joining. So molecule joining, you require DNA line. Moving to the next question, vaccines are prepared from killed microbes are called inactivated vaccine, attenuated vaccine, autogenous vaccine, yeah, none of this. Vaccines prepared from the killed microbes are called as inactivated vaccines or also called as killed vaccines. Very good. Option A is the correct answer. Very good. So inactivated vaccines means a vaccine that does not replicate and therefore unlikely to revert cause of pathological effect. They contains dead virus, incapable of infecting peoples. Okay, attenuated vaccine means this is the live attenuated vaccine, the vaccine that created by reducing the virulence of the pathogens. Okay, what is autogenous vaccines? So autogenous vaccines are called as autologous vaccines. So they are prepared by isolation and destruction of microorganism infected individuals. Okay. So moving to the next question, traffic flow analysis is a technique associated with manufacturing unit, pharmacy stores, marketing, या drug control department. Traffic flow analysis. Yes, answer this question. Traffic flow analysis is associated with what? Traffic flow analysis is associated with marketing. Okay, marketing it is associated with. Traffic flow analysis techniques commonly associated with marketing. Okay, so in the context, okay, traffic flow analysis refer to the study and analysis of. Abhishek is asking about live vaccines. Okay, so Abhishek, live vaccine is a type of vaccine which basically contains your pathogens which responsible for the causing this disease. But there is a twist. What is that twist? Their immunogenic property is there, but their antigenic property has been blocked. That means they only influence or they basically initiate the immune response inside the microorganisms. Okay, I hope Abhishek you got your answer. Next question. Which of the following is not derived from the alpha ketoglutarate? So you know this TCA cycle, during this TCA cycle, many enzymes or byproducts are involved. From these byproducts can also be produced from different amino acids. The question is, which of the following is not derived from the alpha ketoglutarate? Okay, your options is glutamate, proline, arginine, or leucine. So if you'll see the correct answer is leucine. If you'll see arginine, histidine, proline, and glutamate. These four are responsible for the alpha ketoglutarate. Similarly, for fumarate production, phenylalanine and tyrosine involved. For oxaloacetate, arginine is involved. For pyruvate, your alanine, cysteine, glycine, hydroxyproline, serine, and threonine is involved. Okay, moving to the next question. Virus that can naturally infect the cell and introduce the DNA into the host cell is called as transformation, transduction, conjugation, or electroporation. The virus that can naturally infect the cell and introduce the DNA into the host cell. The virus that naturally infect the cell by which method? Please remember it is by transduction method. Okay, by transduction method. So sometimes the foreign DNA can be packed inside a animal virus and these viruses are naturally infect the cell and introduce the DNA into the host cell. Okay, very good. Option B is the correct answer. Next, active immunity is due to which cell? The active immunity in humans is basically due to which cell? Killer T cell, memory cell, helper T cell, or suppressor T cell? Yes, answer this question. The active immunity is basically due to the memory cell. Please remember, this is due to the memory cells because memory cell basically recall the information about the previous infection. That's why this will provide say active immunity. Okay, so active immunity is due to this memory cell. These are sensitized T cell which retains in the memory of antigen specific for the future. And this active immunity is the introduction of immunity after exposed to an antigen. Because this basically memory T cell recognize the previous infection which is has occurred in the particular person. So moving to the next question, an industry manufacture a chemical by standardized chemical reaction is called as process in the processing industry, analytical industry, extractive industry, yes, synthetic industry. The industry which manufacture chemicals. Okay. The industry which manufacture the chemicals are called as synthetic industry. Okay. So why it's synthetic industry? So synthetic industry, this is an industry that manufacture chemical by standardized chemical reaction is called as synthetic industry. Okay. So moving to the next question, the largest protein in the body. Okay. So one question was asked in GPAT 2000 about largest gene. So largest gene is dystrophin. Largest gene, if question will be there, it will about dystrophin. If it is about largest protein, the correct answer is titin. Okay, titin. Lecture 4 was there in the channel. So lecture number 4 is basically withdraw due to some technical error in this video, which will be uploaded, uh, which we will discuss about the further information. This is basically removed due to some technical error. Okay, so technical error in that video, we have removed that video. Okay, next question. So titin is basically the largest protein, and the question is about largest gene. So dystrophin is the largest gene. Moving to the next question. Southern blot is used for the sequencing of nucleotide probe for hybridization of DNA molecule, hybridization of protein molecule, hybridization of RNA molecule, or hybridization of glycosylated molecule. So hybridization of DNA is done by Southern blotting. Hybridization of protein is done, or detection of protein is done by Western blotting. Hybridization of RNA molecule is done by Northern blotting. Okay, so here the correct answer is option number A, that is hybridization of DNA molecule. Very good. Option A is the correct answer. Next question. For southern blotting, detection of DNA, northern blotting, detection of RNA, western blotting, detection of protein. Next question. Exogenous supply of antibodies provides which of the following type of immunity? Artificial active immunity, natural active immunity, or passive? Exogenous supplied antibodies. So they will provide antibodies. When we talked about antibody, that will provide passive, whether it is a natural passive or it is a artificial passive. So if the question is about natural passive, the correct answer will be your IgA. That means immunoglobulin A, which is basically secreted from the from mother to the offspring through the milk. So that will comes under the endogenous or natural passive immunity. It will, as it is the question is there about exogenous supply, your correct answer will be passive immunity. The correct answer is passive immunity. Okay, natural immunity is produced by antibody made from the exposure to the infection. Artificial will be produced through the vaccination. Natural passive immunity products to the mother's milk. Artificial through the antibodies. Yes, we will talk to our team. We will try to recover that video. Okay. Next question. What is the another term for the day book in accounting? What is the another term for the day book in the accounting? Ledger, sales journal, cash flow statement, या income statement. Another term for day book in accounting is your called as sales journal. Okay, it is called as sales journal. Okay, so day book accounting, it is also called as sales book journal. Okay, next, alpha amino acid react with neem hydride to give which color? Yellow, pink, black, green. This is neem hydride reagent for the detection of the protein. Why it is you, or it is basically given which color? Yellow, pink, black, green. It will give pink color. Neem hydride, I will remember like this. NP. Neem hydride reagent will give pink color. Okay. So acid amino acid with neem hydride, it will produce keto acids, ammonia, carbon dioxide, and hydranten. And this hydranten will further react with neem hydride, it will gives a human purple, pink to purple, or that is called as human purple color. Okay. Option B is the correct answer. Next question. The enzyme DNA polymerase can work only in 3 to 5 directions. 5 to 3 direction, 5 prime to 5 prime direction, या both the direction. So please remember the DNA polymerase enzyme always work from 5 prime to 3 prime direction. 5 prime to 3 prime direction. Next question. So DNA polymerase, this is basically from the 3 prime between the 3 prime hydroxyl group to the 5 prime hydroxyl, and the direction will be 5 prime to 3 prime direction. Next question. The complement fixation test, Wassermann test is done for tuberculosis, hepatitis, syphilis, or typhoid. The complement fixation test and Wassermann test is done for the which disease? Tuberculosis, hepatitis, syphilis, or typhoid. The correct answer is syphilis. For the syphilis detection, this complement fixation test and Wassermann test is used. Okay, the Wassermann test that is used for the diagnosis of syphilis. And question is always asked about what is the positive organism for syphilis. The positive organism for syphilis is Treponema pallidum. Please note it down. The positive organism is for Treponema pallidum. Okay, so moving to the next question. If a customer is allowed to draw a check even if there is no balance in his account, is called as cash credit, overdraft, loan, or bond. If a customer is want to draw check even if there is no balance in his account, what we will call cash credit, overdraft, loan, or bond. We will call it as a overdraft. Okay, we will call it as a overdraft. So overdraft basically allows a account holder to continue withdrawing money even when account has no funds. The overdraft is a financial arrangement between bank and account holder that allows a account holder to withdraw the money than they have in their account. Moving to the next question. Which of the following tests basically describe the plasmid? Best describe plasmid: linear DNA molecule, double stranded RNA molecule, double circular self-replicating DNA molecule, mitochondria. So when we talk about plasmid, so plasmid is a double stranded circular DNA molecule. It is nothing but this is a double stranded circular DNA molecule. Option C is the correct answer. So plasmid is nothing but they are double stranded. This is a small extra chromosomal double stranded circular self-replicating DNA molecule. The plasmids are the most commonly used bacteria for the cloning vector, and its size is around 1 to 500 kilobase pair. Widely used for the genetic engineering purposes, commonly for the prokaryotics. Okay, so moving to the next question. Three-dimensional arrangement of protein is expressed in three-dimensional arrangement of protein is expressed in primary structure, secondary structure, tertiary structure, yeah, quaternary structure. So three-dimensional arrangement of protein is basically the tertiary structure of the protein. It is basically nothing but the tertiary structure of the protein. Okay, so primary structure is a simple linear structure. Secondary structure is the most twisted one. Tertiary structure is the tertiary structure is nothing but this is basically called as your three-dimensional structure, and quaternary structure is nothing but when two or more subunits join together, then it is called as quaternary structure. Moving to the next question. The cheek test is used to determine the susceptibility to which of the following disease? Tuberculosis, diphtheria, scald fever, or brucellosis. Cheek test is used to determine the susceptibility to which of the following disease? Tuberculosis, diphtheria, scald fever, or brucellosis. Yes, cheek test. Yes, cheek test is used to which of the following disease? The tick test is used for the diphtheria. Okay, it is used for the diphtheria. It is used for the diphtheria. Moving to the next question, cheek test is for the susceptibility of diphtheria. Dick test is for the susceptibility of scald fever. Tuberculin test is for the detection of the tuberculous bacillus. Scratch skeleton test for the diagnosis of scald fever. Okay, Dockery test for determining the sensitivity of thermophilus structurally. Next question: the test help to measure the candidate's capacity to learn a particular skill and its potential ability is called as intelligence test, aptitude test, proficiency test, या personality test. This is basically called as an aptitude test. Please remember. Okay, the person's ability to learn a particular skills is basically called as aptitude test. So that's why in your different competitive exam, you will have a aptitude questions. So this is basically evaluate to a person how much they have a capacity to learn. Next question. Dash is the which is the starting material for ketogenesis? Which of the following is a starting material for ketogenesis? Acetyl coenzyme है, acetoacetate, acetoacetyl coenzyme है या acetophenol? The starting material for ketogenesis is your acetyl coenzyme. Please note it all. Starting material for ketogenesis is your acetyl coenzyme A. Okay, option A is the correct answer. Acetyl coenzyme A when react to form acetoacetyl coenzyme A, and that acetoacetyl coenzyme A react with beta hydroxy beta methyl coenzyme A or HMG coenzyme A to form acetoacetate. Okay, so this is basically ketogenesis process. Next question: which of the following enzyme is used to join bits of DNA? So whenever with the you heard the word join, so joining is basically done by enzyme DNA ligase. Okay, joining is done by which enzyme? That is your DNA ligase. Okay, joining is done by enzyme that's name is DNA ligase. Okay, so moving to the next question. Which immunoglobulin can cross? Very good. Option A is the correct answer. Very good. Which immunoglobulin can cross the placenta to provide passive immunity? So already I'm starting, I told you there is an immunoglobulin which can cross the placenta through which it will transfer from mother to fetus and provides immunity. That particular immunoglobulin name is what? IgA, IgD, IgE or IgG? Yes. Which immunoglobulin can cross placenta? Your immunoglobulin IgG, it can cross the placenta. Okay, IgG is the only antibody that can cross the placenta. Okay, it provides a passive immunity to the offspring. Next question. Buffer stock is the level of stock, half of the actual stock, maximum stock in the inventory, minimum stock level below which actual stock should not fall below the reader level. So the buffer stock is nothing but the minimum stock level below which the actual stock should not fall. Okay. So this is basically buffer stock. Synthesis of urea takes place exclusively in. This question is very important. Many times this question I've learned. Synthesis of urea takes place in which of the following organ? Kidney, liver, gallbladder, या urine bladder. Synthesis of urea basically takes place exclusively in, yes, two things you have to remember. Synthesis takes place in liver, elimination. Elimination is done by kidney. Okay, synthesis in liver and elimination or excretion is done by kidney. Okay, that is called as your urea cycle. Next question. Presence of viable bacteria in the blood stream is called as what? Viremia, septicemia, bacteriemia, or bactericidal. The presence of bacteria in the bloodstream, what it is called? Hyrissemia, septicemia, bacteriemia, or bactericidal. So this is called as, yes, bacteremia. Okay. Presence of viable bacteria in the bloodstream is called as bacteremia. Okay. It is called as bacteremia. So bacteremia is defined as the presence of viable bacteria in the bloodstream, and this can occur in the daily activities like toothbrushing and some of the minor medical procedure like dental work, but also during some infection. So classification of bacteria includes primary bacteremia and secondary bacteremia. And septicemia means this is a microbial infection that basically infect the blood that produces illness. Next is toxemia that release the bacterial toxin into the blood. Next question. Which type of ELISA is used to measure the presence of antigen directly? Indirect ELISA, sandwich ELISA, competitive ELISA, yeah, Westermann test. Which type of ELISA is used to measure the presence of antigen directly? Your correct answer is C. Indirect ELISA, you cannot detect the antigen directly. So please remember this option C is the correct answer. Competitive ELISA. This is the most sensitive and most effective, and it directly detect the antigen present in the body. Okay. So sandwich ELISA, this is just specifically designed directly to measure the presence of quantity of antigen in the sample. Okay. Moving to the next question. Which of the following fatty acid is essential? Stearic acid, butyric acid, aristonic acid, or oleic acid? So essential fatty acid means what? Essential fatty acids are the type of fatty acid which basically cannot synthesized in the body. It has to be supplied from the outer source. Okay. So it has to be supplied from outer source. So there are three. One is your linoleic acid, linolenic acid. Another one is your arachidonic acid. These three are comes under your what? These three will comes under your, yes, these three will comes under your essential fatty acid. Okay. So three major are essential fatty acid, arachidonic acid, other is linoleic acid and linolenic acid. Next question. The reproduction rate is equal to death rate. So when you have analyzed the bacterial growth curve, you have seen that. So when that is a phase. Okay. So one is your lag phase, one is your log phase, one is your stationary phase, one is your decline. This is growth and this is your death. Here growth is high, here growth is maximum, here growth equal to death. So in this phase or stationary phase, in this stationary phase, growth, because in that stage your supplement that you have given to the media has been significantly reduced, as well as your what we can call it is significantly reduced, and that's why the reproduction of the bacteria is reduced and death is start increasing. So that's why at this phase growth and the what we can call growth as well as your death is at equal phase. Next question. In DNA replication, the newly added nucleotide joined by growing DNA strand. So which enzyme is involved? DNA polymerase, DNA ligase, restriction endonuclease, or reverse transcriptase. The enzyme that is added to the new nucleotide to the newly strand. So that particular enzyme name is DNA polymerase. Please note my word. So DNA polymerase is an enzyme that basically add newly formed nucleotide to the newly formed strand and helps in synthesizing a new strand. So option A is the correct answer. Very good. Option A is the correct answer. Just give me 1 minute. Okay. So this DNA polymerase is the enzyme that basically catalyzes the connection of nucleotide to form a complementary DNA strand in 5' to 3' direction. The enzyme which catalyzes this joining by newly added strand basically formed to the growing strand. Okay. So moving to the next question. Which of the following RNA structure is similar to the clover-leaf model? Many times this question asks, which RNA? hnRNA, tRNA, mRNA, yeah, rRNA. Which RNA structure is resemble to the clover-leaf model? So if you remember the structure of tRNA, so there is a different side, acceptor side and anticodon side, and that is basically resemble with the clover leaf model. So tRNA basically resemble with the clover leaf model. You can see this, the tRNA basically resembles with the clover leaf with the four arms. So tRNA basically delivers the amino acid for the protein synthesis. The structure of tRNA depicted in the structure resembles the clover leaves. Okay. So moving to the next question, the test that is used to detect the typhoid fever. Yes, answer this question. Very, very important question. Typhoid, how you can detect? So typhoid, this is basically nothing but this is a disease which is caused by a bacteria called Salmonella type. How you are going to detect? Yes, how you can detect this test? Yes, the correct answer is yes. Very good. Option number C, that is rose water. Sorry, detection of typhoid. Okay, typhoid detection by Widal test. Very good. The rose water test and ELISA test. This is for the, yes, here this is a complete list. Rose water test for the detection of rheumatoid arthritis. ELISA and Western blotting. This is used for the detection of AIDS. Okay, it is used to detection of AIDS. Widal test and tube agglutination test for typhoid. Next question: protein that are detected by which technique? So already last previous question we have discussed for the detection of protein. Which technique we use? Yes, very good. Option number B. That is Western blot technique. Western blot technique. By this technique, we basically detect the proteins. Okay. In Southern blotting, we detect DNA. Please remember. Next, Southern blotting, DNA, Northern blotting, mRNA, and Western blotting, protein. Next question, the distance between each turn in the helical strand of DNA is. So you will see, if this is the strand, distance from this to this. Distance from this to this is how much? That is 3.4 nanometer or 34 Armstrong. Okay. 34 Armstrong or 3.4 nanometer. Option B is the correct answer. Okay. Each turn contains 34 Armstrong or 3.4 nanometer. Moving to the next question. Which immunoglobulin is predominantly secreted in human milk? IgG, IgA, IgM, IgG, IgE. Yes. Which immunoglobulin is predominantly secreted by males? The correct answer is IgA. That's why IgA is called as secretory antibody. Already I have told many times. It is called as, it is present in saliva. It is present in saliva also. Okay. Not only in uh, milk, it is also present in saliva also. Okay. Moving to the next question. Screening test for AIDS is done by Western blot test, ELISA test, both A and B, या VDRL test. Screening test for AIDS is done by. So I mean, 2-3 question back, I told you Western blot test technique and ELISA both are used for the detection of AIDS. That is called, AIDS is caused by which virus? Acquired immunodeficiency virus. Acquired immunodeficiency virus. Okay. So moving to the next question. Isoelectric pH of pepsin is 4.6, 1.1, 4.7 and 11.0. So first you remember what is isoelectric point. So isoelectric point is a basically point where the protein basically or amino acid exerted as a deuterant form. That is both positive and negative charge will be there in their uh, uh, structure. So isoelectric pH of pepsin is 1.1. At 1.1 pH, it will show the isoelectric point. So for pepsin, it is 1.1. For casein, it is 4.6. For human albumin, it is 4.7. For urease, it is 5.0. For hemoglobin, it is 6.7. For lysozyme, it is 11. Next question. The presence of mitochondria in the living cells can be determined by the use of. The presence of mitochondria in the living cell can be determined by the use of neutral red, methylene blue, Janus green, or phenolphthalein. The presence of mitochondria in the living cells can be determined by. For the mitochondria, there is a specific dye that is used that is called as Janus green. Many times this question have asked, so please remember Janus green. Janus green is basically used for the detection of mitochondria in a particular living tissue. Okay. So JGB, we also called it as a JGB, Janus green B. Okay, this is a dark green or black powder-like color that basically used to detect the structure of mitochondria in a particular cell. Now, next question. Which of the following enzyme is commonly used in ELISA? DNA polymerase, horse radish peroxidase, restriction endonuclease, yeah, RNA ligase. So if you remember, enzyme ELISA is the full form of ELISA is enzyme linked immunosorbent assay. So this is the techniques where enzymes has been linked to a particular antibody, so which will produce a detect a specific antigen. So most common and the most frequently used enzyme for your ELISA technique is your HRP, that is horse radish peroxidase. Yes, option B is very good. Option B is the correct answer. Horse radish peroxidase is the common correct answer. Next question. Enzyme-linked immunosorbent assay. This is a sensitive technique that use the enzyme antigen antibody combinations. Okay, so this is a biochemical technique that is mainly used for the immunological function. So most commonly used horseradish peroxidase. Other than that, alkaline phosphatase, beta galactosidase can also be used. Next question. The functional enzyme is referred as apoenzyme, coenzyme, haloenzyme, proenzyme. The functional enzyme, the functional part of the enzyme, because enzyme has two parts, uh, protein part and non-protein part. The functional part of the enzyme is called as basically holoenzyme. Okay, holoenzyme. So holoenzyme, they contain both your protein part as well as non-protein part. Please remember, holoenzyme contain your both protein as well as non-protein part. Moving to the next question. The process of eliminating the plasmid from the bacteria is known as eliminating the plasmid from the bacteria is known as episome, conjugation, transduction, yeah, curing. Yes, the process of eliminating plasmid from the bacteria is called as what? Episome, conjugation, transduction, yeah, curing. The correct answer is curing. Okay, please remember the correct answer is curing. So the process from which a plasmid can be removed is called as curing. Next question. The communicable disease caused by RNA paramoxyvirus is chickenpox, measles, tuberculosis, or diphtheria. The communicable disease caused by RNA paramoxyvirus. Chickenpox, measles, tuberculosis, or diphtheria. Yes. Okay. The communicable disease caused by RNA paramoxyvirus is your measles. Okay. Measles is the correct answer. Next, the paramoxyvirus. This is a family of single-stranded RNA virus that basically associated with the disease called measles virus. Okay. So moving to the next question. Which of the following equilibrium suggests the non-competitive inhibition of enzyme for the conversion of substrate to product with inhibitor? Yes, sir. खाना खा के आऊं ऐसा तो कोई सर बोला नहीं सर मैं पूछता हूँ काम कर रहे हैं आप में कर रहे हैं तो वो लोग थोड़ा करते हैं तो नहीं मैं तो नहीं आऊंगा सिर्फ खाना खाने के बाद वो आते हैं मैं भी नहीं मैं तो आज जाने वाला हूँ अभी से खाना खाने आया था मैं अभी आऊंगा सर मतलब 9:30 तक यस Okay. So the correct answer is option number B. Okay, you will see. So when enzyme plus substrate react, there will be a formation of enzyme substrate complex, and there will be the final product is an enzyme plus product. And when the enzyme plus product is formed, the next the enzyme will be released, and the enzyme will again further react with other compound to form the product. So this is the way how this product is. Okay, so moving to the next question, the organism which used vitamin B12 fermentation is called as Saccharomyces cerevisiae, Pseudomonas denitrificans, 𝙂𝙑𝑃𝑆, and Lactobacillus delbrueckii. So the organism that use vitamin B12 fermentation is basically nothing but your Pseudomonas denitrificans. This is nothing but your Pseudomonas denitrificans. Please note it down. Pseudomonas denitrificans, this is basically used for the production of vitamin B12. Many times this question I've asked in exam. Please note down. Moving to the next question. The antibody that is capable of neutralizing the exotoxin is called as endotoxin, epitoxins, antitoxin, or serotoxins. The antibody that capable of neutralizing exotoxins are endotoxin, epitoxin, antitoxin, or serotoxin. Yes, the correct answer is antitoxin. So basically, antibody can be neutralized by antitoxins. So antitoxins are the antibody that is made against the microbial exotoxin. It is made against the microbial exotoxin. Okay. So moving to the next question, the codon AUG is nothing but chain initiating codon, release of factor for peptide chain, recognition site for tRNA, chain termination. So AUG is nothing but this is a basically in start codon. This is nothing but called as start codon. Okay, this is in when this codon binds to a DNA strand, it will start the initiation process of uh, chain elongation process of DNA strand. Okay, so this is nothing but this is a initiation codon. Okay, moving to the next question. Other than the AUG, you have GUG. GUG also a initiation codon. Next question. Which of the following is the final electron acceptor of the lactic acid fermentation? Okay. So you know lactic acid fermentation in which this is a type of fermentation technique. So this is so what will be the final electron acceptor? Lactic acid, oxygen, pyruvate, yeah, NAD. What is the correct answer? The final electron acceptor is nothing but the pyruvate molecule. Okay, pyruvate is considered as the final electron acceptor. It is considered as the final electron acceptor in case of lactic acid fermentation. Okay, so in lactic acid fermentation, the glycolytic product pyruvate is reused to the final electron acceptor and regenerate the NADH molecule. Yes, option C is the correct answer. Very good. Option C is the correct answer. Okay, so moving to the next question. Varicella vaccine is given for the prevention of which of the following disease? Tuberculosis, chickenpox, cervical cancer, or hepatitis B. Varicella vaccine. VZV virus, varicella zoster virus that is responsible for the cervical cancer. That's why VZV or varicella vaccine is basically given for the cervical cancer. Yes, answer this question. The correct answer is okay, okay. Okay, VZV is nothing. This is that is different. VZV virus different. So varicella vaccines. This is caused by varicella zoster virus. it is caused by itchy rashes, like you can see fluid accumulation in the skin. Okay. So that is basically, this is a causative organism. Okay. This is a causative organism for the chickenpox virus. Moving to the next question. DNA and RNA contains following two major purine bases. DNA and RNA contains two major purine bases, which includes 196 cytosine. So you will see nitrogenous bases. There are two types. One is your urine, another one is your pyrimidine. Purine is basically your adenine, guanine. Pyrimidine is thiamine, cytosine, and uracil. Three major pyrimidine. So if we talk about nitrogenous bases of purine, that is ATGC present in DNA. If we talk about RNA, it is ATCO. Okay, so already it has been there. So in DNA, it is adenine plus guanine, and pyrimidine base is thiamine plus cytosine. In RNA, it is adenine plus guanine, pyrimidine base is uracil and cytosine. Please remember. Next question. The organism that is used for the production of dextran is Leucansta mesenteroids, Lactobacillus vulgaricus, Clostridium botulinum, yeah, Gamilobacter botulinum. So if you will remember, I have explained you in the class that, so what is organism that used for the production of dextran? That is nothing but your, yes, answer this question. The correct answer is leucoconstruct, leucoconstruct mesenteroid. Please remember, there is a, this, you can follow this table. This table contains all the information. If for the ethanol production you require Saccharomyces cerevisiae, for lactic acid production you require Lactobacillus vulgaricus, for the enzyme alpha amylase production you require Bacillus subtilis, for dextran production you require leucoconstruct mesenteroid, which was there in our options. Okay. For the lipase enzyme, we require Candida lipolytics. For penicillin, we require Penicillium chrysogenum. For streptomycin, we require Streptomyces griseus. For chloramphenicol, we require Streptomyces benzoyli. For riboflavin, we require Ascorbyl. For vitamin B12, we require Pseudomonas dentificus. Moving to the next question, which of the following is responsible for the development of cellular immunity? Your options are T lymphocyte, NK cells, neutrophils, या B lymphocyte. Which of the following is responsible for the development of cellular immunity? T lymphocyte, NK cells, neutrophils, या B lymphocyte. Cellular lymphocyte. If we talk about humoral immunity, what will be the answer? The answer will be B lymphocyte. When we talked about cellular immunity, our answer will be yes. Yes. What will be the answer? Yes. What will be the answer? The answer will be yes. T lymphocyte. T lymphocyte, na? Okay. Next question. Next is T lymphocyte. Okay. Antioxidant vitamin is, which of the following is an antioxidant? Vitamin K, vitamin D, vitamin E, and vitamin B6. When we called about antioxidant, so antioxidant also called as anti-sterility, antioxidant or anti-sterility vitamin. That is nothing but your vitamin E. That is nothing but your vitamin E. Okay. Next question. Vitamin E is also called as tocopherol. So this is called as vitamin in search of disease also. So Evans and his associate has first time identified this vitamin E. Okay. Next question. Micropropagation of the plant is carried through. Micropropagation of the plant is carried through cross fertilization, seed germination, plant tissue culture, and grafting. Okay. Cross fertilization, seed germination, plant tissue culture, and grafting. What will be the correct answer? Micropropagation. So micropropagation is a technique in which we are basically produce a hybrid plant. So this is basically done by a technique called plant tissue culture. This is done by a technique called as plant tissue culture technique. This is done in plant tissue culture. So method for micropropagation includes meristematic culture, embryo culture, callus culture, protoplast culture, as well as suspension culture. So micropropagation is nothing but this is the artificial process of producing a plant vegetatively through the tissue culture or cell culture technique. Okay, so moving to the next question. Which of the following method of sterilization is based upon the coagulation of the protein? Which of the following of sterilization is basically based on the coagulation of the protein? Dry heat, moist heat, radiation, or gaseous sterilization? Which of the following is called as sterilization based upon the coagulation of the protein? Yes, which type of sterilization? If you'll remember, we discussed very detailedly about moist sterilization. So moist sterilization is nothing but so that is nothing but the moist sterilization. Okay, so in moist sterilization, the major principle involved is your coagulation of protein. So in dry heat, that is thermal activation or oxidation of the cells. In moist heat, that is coagulation of the protein. In radiation, that is destruction or disorganization of enzyme or DNA. In gaseous sterilization, the technique is alkylation. In the filtration, that is physical separation of microorganisms. Next question. SGPT is elevated in hepatitis, diabetes, pancreatitis, none of them. So I have taught you two important enzymes. One is your SGPT, one is your SGOT. One SGPT, that is serum glutamate pyruvate transaminase, SGOT is serum glutamate oxaloacetate transaminase. These two enzymes are severely elevated in case of which disease? Liver disease. Nothing but in case of liver disease. Option A, that is hepatitis, is the correct answer. Okay, alkaline transaminase or SGPT basically inhibited in case of acute hepatitis. Moving to the next question. The method that is effective for producing virus-free plant is root culture, meristem culture, somatic embryogenesis, or floriculture. The most effective method for producing virus-free plant is, yes, answer the question. We have discussed many times about this question. Which is produce the virus-free plant? Very good. Option B, very good. Option B is the correct answer. That is your meristem culture. Meristem culture is nothing but this is considered as the what? The method through which virus-free plant can be produced. Sometimes you know a virus can attack the plant, which can affect the productivity of a plant. So through the meristematic culture, we can produce a virus-free plant. Moving to the next question. The pathogenic organism in milk is killed by kindalization, pasteurization, autoclaving, or dried chillation. I hope all of you are aware with this question. So this pathogenic microorganism that can kill by technique, that technique is particularly called as pasteurization. Which technique it is called? It is called as pasteurization. The pasteurization is the technique through which the, the pathogen that present in the milk can be killed. So, okay. So that is by two major method. One is your Holder method, another one is your flash method. In Holder method, you have to 63 degree for 30 minutes you have to boil, or in case of flash method, 72 degree per 20 seconds. So this basically kills the non-pathogenic sporic microorganisms such as mycobacteria, Salmonella, like organisms. Moving to the next question. Calcitriol each 125 dihydroxycholecalciferol, 25 hydroxycholecalciferol, 24 25 dihydroxycholecalciferol, yeah, 1 OH cholecalciferol. What is calcitriol? Calcitriol, this is nothing but it is 125. This is the most potent form of vitamin D. It is the most potent form of vitamin D, and it is nothing but 125 dihydroxy cholecalciferol. Okay, so moving to the next question. Which among the following is the first transgenic plant? First transgenic plant. Yes, tobacco, senna, aloe vera, ya clove. First transgenic plant. Yes, answer this question in the chat box. Which of the following is the most first transgenic plant? The first transgenic plant, yes. So the first transgenic plant that is your tobacco, and the virus that infect that plant is that is called as TMV. That is tobacco mosaic virus. That is nothing but your tobacco mosaic virus. Moving to the next question. HEPA filter prevents the entry of microbes. HEPA filter prevents the entry of microbe of size less than zero point 0.3, less than zero point 0.1, more than zero point 0.3, more than 0.1. HEPA filter prevents the entry of microbes with the size, yes, more than 0.3. Very good. Option C is the correct answer. HEPA filter basically prevents the entry of microbes of size more than 0.3 micron. Okay. C. More 0.035 micron, and its efficiency is 99.997%. UPA filter having efficiency 99.97%. Particle size up to 0.1 micron. Next question. Deficiency of biotin leads to dermatitis, anemia, both A and B. Yeah, very. So biotin deficiency. Biotin deficiency, yes. Vitamin B7 deficiency. So vitamin B7 deficiency, very, yes. Answer this question. Vitamin B7 or biotin deficiency that can causes the major symptoms. Please remember, major symptoms is dermatitis, but it can also causes anemia. Okay, anemia is not prominent, but the major option is dermatitis. So here both option, but only if anemia is not there, dermatitis only is there, then dermatitis will be correct answer. Okay. So itching as well as anemia also can occur, but the anemia is not prominent. Please remember dermatitis is more prominent. Haploid plants can be obtained from anther culture, bud culture, leaf culture, or root culture. Yes, answer the question number 77. Haploid plants can be obtained from, yes, haploid plants. Please remember option A is the correct answer. Anther culture. From the anther culture, we basically obtain this anther culture. So from this anther culture, we are basically get haploid man. That means so we will get so haploid plant anther culture or androgenesis, that is the production of haploid plant from the microspores. So anther culture production haploid reported about 250 species. So haploid plant can be produced by anther culture or microspore culture. Okay, moving to the next question. Riddle-Walker test is performed by using which test? Salmonella E. coli, Salmonella Nuris, Staphylococcus pyrogens, or Salmonella typhi. Many times this question was asked about Riddle-Walker test. So Riddle-Walker test is nothing but this is a comes under a phenol coefficient test. This is basically measure the efficiency of a disinfectant. So Riddle-Walker test is one of the tests. So they basically use a standard microorganism. What is its name? Which one is correct? Yes, answer this question in the chat box. E. coli, Staphylococcus neruri, Staphylococcus pyrogens, yes, Salmonella type. Yes. Yes, option D is the correct answer. Salmonella typhi, please remember this is the gold standard. Okay, this is the gold standard. This is used for the why it is used. This Salmonella typhi, this is a gold standard. This is used for the perform for the test of Riddle-Walker test for to detect the efficiency of a disinfectant. Moving to the next question. Normal SGOT level in the serum is 8 to 45 units, 80 to 100 units, less than 8 unit, yeah, 8 unit. So please remember, SGOT, SGOT limit is 8 to 45 international unit. SGPT is 8 to 40 international unit. SGOT is 8 to 45 international unit. SGPT is 8 to SGOT is 8 to 45 international unit. SGPT is 8 to 40 international. Moving to the next question. Which among the following chemicals are the most widely used for the protoplast fusion? Very, very important question. Many times this question was asked. For the protoplast fusion, which chemical is basically used? Okay. So your option is mannitol, polyethylene glycol, sorbitol, or manol. So the chemical that is most widely used for the protoplast fission is nothing but your polyethylene glycol. Please remember PEG, polyethylene glycol. Many times this question has protoplast fusion. For the protoplast fission, PEG. Sometimes in option PEG also there. So PEG if there, PEG means polyethylene glycol. So you have to select that PEG. Mannitol, sorbitol, manol, these are the sugar derivatives. Okay. Protoplast fusion it is used. Next question. The principal structural component of the cell wall in the bacteria is made up of complex polysaccharide, simple protein, glycoprotein, or peptidoglycan. Principal structural component of the cell wall. Yes. Answer this question in the chat box. Yes. Option D. That is peptidoglycan. It is synthesized from the peptidoglycan. Okay. These are the cell wall that is rigid in structure made up of peptidoglycan that surround the plasma membrane as an external. Okay, and that is consisting of N-acetyl mumaric acid and N-acetyl glucose. Moving to the next, achloric acid. This is a toxic jaundice, hemolytic jaundice, amoebic dysentery, obstructive jaundice. So, you will talk about jaundice. Yes, answer this question. Achloric acid. This is a type of hemolytic jaundice. Okay, this is a type of hemolytic jaundice. Achloric jaundice, this is basically without bilirubinemia, which is associated with elevated unconjugated bilirubin. It is associated with elevated unconjugated bilirubin. This is nothing but associated with familial achloric jaundice, also called as hemolytic jaundice. Okay, so here you can see there is white piece of eye turn into yellow. You will have a yellow skin. You can see this is in normal cases. Moving to the next question, which vaccine is produced by recombinant DNA technology? Tetanus vaccine, poliomyelitis vaccine, hepatitis B vaccine, या toxoid vaccine. Which vaccine is produced by RDT, recombinant technology vaccine? Yes, yes. Answer this question. So the vaccine that is produced by recombinant DNA technology is nothing but that is your poliomyelitis vaccine. Okay, poliomyelitis. Sorry, hepatitis B vaccine. Poliomyelitis vaccine not. It is hepatitis B. This is also called as third generation vaccine also. Okay, so sorry, second generation vaccine. The vaccine that prevent the hepatitis B and it is produced by the yeast. So and this plasmid is formed from the recombinant. Okay. What is the cluster of polar flagella called? Peritrichous, monotrichous, amphitrichous, lophotrichous. So on the based upon the flagella, the bacteria are divided. They are monotrichous, lophotrichous, peritrichous, or amphitrichous. The cluster of polar flagella is called as peritrichous, monotrichous, amphitrichous, or lophotrichous. Cluster of flagella. When we word this word cluster, so lophotrichous means there is cluster, that is group of flagella at a site. Okay, so you can see cluster of flagella is called as lophotrichous. So lophotrichous bacteria have a dwarf flagella at one or both of it. Okay, so you can see cluster of flagella at single end or multiple. Next question, vitamin B2 is precursor for coenzyme pyridoxal phosphate, vitamin B2, one that is riboflavin. Riboflavin is a precursor for pyridoxal phosphate, coenzyme thiamine pyrophosphate, FMN, या NADP. Ribotal, that is riboflavin, is a basically coenzyme for FMN and FAD. Okay, thiamine is a coenzyme for the thiamine pyrophosphate. Niacin is coenzyme active form is NAD and NADP. Okay, you have to remember from this table, you can 100% you will see a question this year. Okay, so what is the active form and what is the ring present in it? Okay, what is the active form and what is the ring present in it? Moving to the next question. ELISA, that is enzyme linked immunosorbent assay, allow the rapid screening and application of the presence of dash in a sample, amino acid, DNA, antigen, या protein. The ELISA enzyme linked immunosorbent assay allow the rapid screening of. Yes, answer the question number 86. ELISA is basically used for the detection of, yes, antigen. ELISA is a antigen antibody detection technique. No, it is a antigen antibody detection technique. Okay, this is nothing but this is antigen antibody detection now. Next question, which of the following is a major features of spirochetes? Rigid and polar flagella, flexible or periplasmic flagella, soft and non-motile, rigid and lacking flagella. Which of the following is a major features of spirochetes? Rigid and polar flagella, flexible or periplasmic flagella, soft and non-motile, rigid and lacking flagella. Yes, spirochetes. What is the feature of spirochetes? Spirochetes having special feature that is flexible in the periplasmic flagella. Flexible and periplasmic. Okay, option B is the correct answer. Next question. Anaerobic dehydrogenase, sorry, aerobic dehydrogenase is a biological oxidation contains NADP, FAD, NAD, FMN and FAD or NADPase. Aerobic dehydrogenase in biological oxidation contains what? NADP, NAD, FMN and FAD, yeah, NADH. In the enzyme aerobic dehydrogenase, in biological oxidation is nothing but this is a process in which our production of, or this is a cellular oxidation process in through which ATP is actually produced. We know that during this glycolysis with TCA cycle, ATP is produced. But this is the actual process by that which is called as biological oxidation through which your what we can call actual ATP will be produced here. So that is what it contains. It basically contains FMN and FAD. Please remember FMN and FAD content. Yes. Answer option C is yes. Option C is the correct answer. Very good. Option C is the correct answer. So they are flavoprotein enzyme having FMN and FAD in the structure. Okay. So these are the catalyzed removal of hydrogen from the substrate and use their oxygen for the artificial substance such as methylene blue as a hydrogen gas. Moving to the next question. Organism having optimum growth temperature is greater than 45 are called as mesophile, thermophile, psychophile, monophile. So based upon the temperature, please remember, based on temperature, bacteria are divided in three classes. One is called as psychophiles, mesophiles, and thermophiles. When the question is about temperature above 45, that is called comes under your thermophile class. The temperature around 45 degree, more than 45 degree. Optimum growth temperature above 45 degrees. So bacteria like Bacillus thermophilus, Archegallus, and Thermophilus aquaticus. Archeglomus. Next question. Cytochrome oxygen contains copper and zinc. So that is called as metallozyme. You heard this term metallozyme. So metallozyme means the enzyme contains metal as a substrate. Okay. So it contains cytochrome contains copper and zinc and iron, copper and iron, yeah, manganese and copper. Yes. Answer this question in the chat box. Cytochrome oxidase contains, yes, option C, very good. Copper and zinc in its structure. It contains cytochrome oxidase, copper and iron in structure. This question was asked in GPAT 2016. Xanthine oxidase contains molybdenum. Pyruvate oxidase contains manganese. Phenol oxidase contains copper. ATPase contains magnesium and calcium. Enolase contains magnesium. Moving to the next question. All of the followings are the simple RNA virus except. All are simple RNA virus except rubella virus, parvovirus, rabies virus, या polio virus. The question is which of the following is not a RNA virus? If I'll simplify the question which of the following is not a RNA virus? That is yes, rubella virus. Option A. Rubella virus, it is not a RNA virus. It is a parvovirus. Sorry, parvovirus. Parvovirus is nothing. This is a DNA virus. Okay, this is not a RNA virus, this is a DNA virus. You can see parvovirus, this is a single stranded DNA virus. But if you talk about RNA, your Picrona, Corona, Toga virus, Guinea virus, Orthomaxi virus, Rhoda virus, and Filovirus, all these are comes under your RNA virus family. Moving to the next question, when accurate determination of glomerular filtration rate is required, which clearance test is preferred? So this is question is about what is used to measure the clearance test to measure the glomerular filtration rate. Creatinine, urea, inulin, ammonia. Yes. Option C, very good. Inulin, this is a polysaccharide. This is a polysaccharide which is used for the accurate determination of GFR. So can you comment what is the normal GFR value for humans? What is the normal GFR value for humans? What will be the value? Then we called it as a, yes, that particular person is healthy. Yes. Come, put down your answer in the chat box. What is the normal GFR value for a healthy humans? Put your answer in the comment. Sorry, in comment as well as in the live chat session. So normal value is, yes, 120 ml per minute. Okay. The normal clearance value for inulin is 120 ml per minute. Next question. Varicella zoster is the positive organism for smallpox, dermatophytes, herpes, yeah, all of this. Varicella zoster. Previously, such type of similar question I have explained you in the previous question. So please read the question and give the answer carefully. Yes, answer this question.
Let's say you have a newspaper in your hand.
Yes, answer this question. 120 ml per minute. Very good. Next question. Varicella zoster virus is a causative organism for herpes. Yes, C. Option C is very good. Option C is the correct answer. Varicella zoster virus. This is a chickenpox virus. It is an causes acute infectious disease. Varicella zoster virus basically comes under herpes virus group. Okay, that basically causes the infection VZ. Okay. Okay. So moving to the next question. The normal urine output in adult 500 to 800 ml per day, 800 to 2000 ml per day, 2 to 3 liter per day, 3 to 4 liter per day. Normal urine output. Normal urine output. Yes, very good. 800 to 2000 ml per day. Okay, 800 to the option C is the correct answer. So in 24 hours, it will be produce 800 to 2000 ml of urine per day. Reduce urine volume by basically caused due to dehydration step. Increase urine volume in the symptoms like diabetes, diabetes insipidus, high fluid intake, and renal abnormality, and when you are consuming diuretics. So in such cases, your urine output can be increased than the normal. Very good. Very good. All of you are given the correct answer. Very good. Okay. So moving to the next question. Phases which show lysogenic cycle are known as lytic phase, tem- temperate phase, virulent phase. Yeah, none of these. The phases which show lysogenic cycle are called as lytic phase, temperate phase, virulent phase, and none of these. Yes, answer the question number 95. 1 to 1.5 liter. Motin Sen, very good, correct. Rajla Sherin, 125 ml per minute. Yes, correct. Yes, yes. For one, just in 2-3 minutes, we will wind up the session. That is your temperate phase. So temperate phase are comes under bacteriophage, which basically display the lysogenic life cycle. Lysogenic, this is a characteristics by an antigen of a bacteriophage. Okay, bacteriophage the only use this lytic cycle called as virulent. The correct answer is temperate phase. Moving to the next question. Ferritin is coenzyme, The stored form of iron, non-protein moiety, या isozyme. So ferritin. So when we heard this ferritin, we recall this is related something to iron. So ferritin is nothing but this is a storage form of iron. Option B is the correct. This is nothing but the storage form of iron. Iron is stored in basically in the liver, spleen, and the bone marrow in the form of ferritin. And in the mucosal cell, ferritin is the temporary storage of the iron. A molecule of apoferritin that combines with 4,000 atoms of irons. Okay. Next question. Which of the following microorganisms multiply by the process budding rather than the fission? Yes. Answer the question number 97. Very good, Aditya. Yes. See, so we have kept such type of question in this revision series so that it will boost your confidence as well as such type of question will 100% ask in your exam. So that's why we have prepared the things like this way. Okay. So yes, fungi. Option B is very good. Option B correct answer. Fungi is the correct answer. Fungi is basically reproduced by budding rather than fission. So budding, fission, and fragmentation, these are taken place by fungi. Moving to the question number 98. The decrease in water content in the body is termed as overhydration, rehydration, dehydration, yeah, both AM. When your body fluid decrease, what we call dehydration, overhydration, rehydration, or all of this. Yes, now answer the question number next and answer the next question. Question number 98. Yes, Pavan C, Rajeshwari C, Raj Trivedi. Okay. Very good. That is called as dehydration. So dehydration is a condition. So in which what happens? So that is when your fluid content reduces. So that might be due to shock, any type of shock. Okay, like hypovolemic shock or any type of burn cases also, your fluid content is reduced. Even in case of diarrhea also, your fluid content is reduced. That's why doctor provides you solution of glucose, lactose, regal lactose solution, dextrose solution we basically provide to increase your fluid content. Option C is the correct answer. So when basically are characterized by water depletion from our body. Okay. So next question. What is the region of decolorization in the gram-negative bacilli in gram state? So why it is basically decolorized? Due to presence of echoic acid, due to presence of cell membrane integrity, due to two dyes used in staining, or due to this lipid content? Yes, answer the question number 99. Yes, answer this question. See, the gram-negative bacteria basically show decolorization due to the lipid content. Okay, so decolorizing agent like ethanol or acetone, they interact with the lipid of the membrane of both gram-positive and gram-negative bacteria that will leads to loss the cell for this peptidoglycan. Last question of the today's session, the normal level of calcium in the blood plasma. This is the last session. ये मैराथन वाली वीडियो हमेशा रहेगी. Yeah, this video will be there. Okay, this video will be there in your our YouTube channel, so you can watch later also. Yes, this video will be there. Answer the last question. The normal level of calcium that present in the blood, it is around to 9 to 11 milligram in 100 ml, or sometimes it is written that 9 to 11 mg per dl. Okay, so correct answer is option A. Okay, so the most of the blood calcium present in the plasma, since the blood contains very little of it. Normal concentration is 9 to 11 mg per deciliter or 4.5 to 5.5 milliequivalent per liter. Okay. So is this clear to all of you? All of you, I hope all of you enjoyed the session. If all of you have enjoyed, please give a thumbs up in the chat box. All of you, please put a thumbs up in the chat box. I will consider it as a all of you have understood. So what will be your suggestion? If you have any type of suggestion, you can give. We will take your consider your suggestion from our next series. So please suggest or please give a thumbs up in the chat box. So we will consider it all of what if you have any type of suggestions, you can also give. Thank you so much for so much effort. Thank you so much for attending. I hope okay, we will consider. So thank you so much for your so much attention in this class. I hope this session, the 5 day session we kept for you will be fruitful in your upcoming GPAT 2025 exam. All the best for your upcoming GPAT 2025 exam. So if thank you so much, Karthik. And this is just a simple reminder that please download your admit card. Please check what are the document you will need because you will have Sunday your exam. So please uh, consider your document. What are the document you need, and please ensure about the what is your exam center and what is the distance between from your staying place to your exam center, and please make sure about all the things will be go uh, so so there is a simple thing in Hindi what we call there is a very common dialogue in that uh, movie 12th fail, I think 12th fail. Okay, so if you want to succeed, like एक पत्थर तो फेंक दो यारो इन आसमान में. So मतलब इसका मतलब is like, so you have to keep your efforts continuing. You have to like we have to make our hard work, dedication, and whatever the things we require, you have to complete it to the 24th night. In the 25th, we have to give our 100%. And one of the most important thing in exam, student they do is when they see the question, difficult question, they basically uh, forget all the things and they don't know and they forget all the things and they will not able to answer a single question and they will do silly mistake. So in that cases, what you will do is always try to focus on the question. Try to be calm in the exam. Take your water bottle. So whenever you feel any stress, have some sip of water. It will help you to concentrate. And very importantly, always read the options when you are giving the question. Read whether there is any exception, except or not, or and or type of option will be there in the exam. Very careful for that. Okay. So that's all for today. All the best for your upcoming GPAT 2025 exam. We are with you from the day one when you start your preparation. We will be with you till you guys reach to a renowned place or renowned goal of your life. We will be there throughout your journey. Whenever you will feel any ID card, jaruri. Yes, definitely. Your ID card is the only thing that will help you to enter into the exam. Okay, that's all.